So it's 1 o'clock, Roberto. So Roberto Lopez is the speaker. He's going to talk about LED lightings. He's at Purdue University. And Roberto's done a lot of research over the last few years, including some work that he did when he was a graduate student at Michigan State. And he's continued that work at Purdue University. So he's our presenter today about LED lighting. So I'll turn it over to you, Roberto, and I'm going to mute myself off.
All right. Well, thank you very much, Brian, and welcome everyone to today's webinar, where we're going to look at basically the potentials of using LEDs, whether it's in the greenhouse or it's in indoor production of ornamentals and leafy green crops. Let's start off with a brief introduction in terms of greenhouse lighting. This is typically what we find with greenhouse growers is that they can take advantage of light, whether it's light quantity, light quality, or light duration. And up until now, we really focused on, in terms of light quality or intensity, using high pressure sodium or metal halide lamps to really give us that photosynthetic lighting to increase that daily light integral in our greenhouse. And ultimately, that affects the plants in terms of roots, shoots, growth, branching, flower number. And then in terms of light quality, really the only thing we could do up until now is have some sort of filter or shading in our greenhouse, which would affect photomorphogenesis or pigmentation. And then, of course, probably one of the most common is photoperiod, where we typically would use an incandescent lamp in terms of inducing flowering. Of course, incandescent bulbs are being phased out of production. Now, emitting diodes, which we can use for photosynthetic, photomorphogenic, and photoperiodic lighting. And I'm going to try to talk about all of those today. I have quite a few slides, and so we may not get to the photoperiodic lighting, but of course you have that material with you. In terms of greenhouse grower challenges, light intensity is obviously a big problem, especially this time of the year. As I look out the window, I can definitely see that supplemental lighting is necessary. And why do we use supplemental light? Well, propagators of young plants are really interested in minimizing the production time, whether they're rooting cuttings or they're producing plugs. When we have low light levels, it typically takes much longer to root those cuttings or to produce a marketable plug. So we really want to use supplemental light. We also want to optimize the quality of those young and finished plants. During this time of the year, especially during peak young plant production, we typically have outdoor photosynthetic daily light integral in the range of 5 to 20 moles across the northern U.S. Obviously, as we go further south, that number increases. However, once we move into a greenhouse, what we often find is that light levels can be 50% or less of outdoors due to the structures, the glazing material, the energy curtains, and of course those hanging baskets. So really, during this time of the year, we can have daily light integrals as low as 1 to 5 moles, and even lower during extended periods of cloudy weather. We have basically a map of the U.S., and we're looking at the light integral and young plant production. So we can see that during peak production, which is this month, across the northern U.S. And even midsections of the U.S., you can see that the benefits of supplemental lighting are quite high. And so, what happens with supplemental light? So we can see that photograph that large propagators definitely use supplemental lighting to increase that daily light integral. Well, this is some of the work that was done by one of my graduate students, and you can see that for both Geranium and Dioecia, as we increase that daily light integral from very low light levels of 1 mole all the way up to about 12.5 moles, excuse me, we obviously increase root growth, root mass, and of course with the Dioecia, we can see that we have an increased number of branches. So what does this ultimately for the grower? Well, you're going to get this plant out of the greenhouse much faster. This is some work that was with plugs. And what basically is, as we can see here, these are plants that did not receive any supplemental light. So this was 27 or 33 days after seed sow, and you can see that the plants are relatively small, very few roots. And then they basically divided the time into thirds. So here this H basically stands for providing supplemental lighting during the first third, the second third, or the last third, or providing supplemental light at the beginning, at the end, or during the entire production of these seedlings. And you can see, obviously, if you provide supplemental light, you're going to get that rooted plug out of the greenhouse faster, which is going to save you an energy cost. When we look at species, they can respond very differently in terms of supplemental light. So you can see that Agaranthemum is very responsive to DLI. As we increase from 1 all the way up to 12.5 moles of light, you can see how that root dry mass increases quite a bit. There's other species such as Angelonia that you can see that right around 7 or 8 moles, really root production starts to level off. We also find that as you increase that daily light integral in the greenhouse, photosynthesis increases. So here we're looking at New Guinea impatiens. So this basically is showing us when those cuttings were under calycine, then they were transferred into different daily light integrals, and you can see those big differences in terms of photosynthesis, which is ultimately going to affect growth. Well, this leads us into our first experiment or study that I'm going to talk about. This is some work that was done by Chris Curry. Basically what Chris did is we wanted to compare rooting cuttings of Geranium, Petunia, and New Guinea impatiens. And we wanted to compare that to LEDs. So we used 3 different LEDs, one that had 100% red light, another that had 85% red and 15% blue, or 70% red and 30% blue. We had a 16-hour photo period where we provided 70 micromoles per meter squared per second of supplemental light. Again, we also had that high pressure sodium treatment, and our air temperature set point during propagation was 73 degrees Fahrenheit. Here we have our spectral output of the various supplemental light main treatments. So you can see here we have our high pressure sodium. Of course, we have peaks in the orange region, which give us that color of the high pressure sodium. But you can see with the LEDs that really we can concentrate on that red or blue region. For example, here we have our 70% red treatment, and then you can see again our peak in the blue region, which is the 30%. Excuse me, we have their blue and our red. What did our plants look like? Well, here once again this is supplemental lighting, so we have sunlight plus high pressure sodium and our various LED treatments. When we look at these rooted cuttings, we see that basically there are little to no differences between whether this cutting was rooted under high pressure sodium or whether it was rooted under LEDs with increasing amounts of blue light. See that again with Petunia. When we look at photosynthetic rates, if we look at the black bars, which are New Guinea impatiens, we can see that basically photosynthesis was the same across the various supplemental lighting treatments. We found this with all 3 species. As you can see, those letters are all the same, indicating that there were no statistical differences. We then took the plants and we put them into a common environment. So after the 28 days of propagation, we moved them into a common environment to see if there were any residual effects of that supplemental light. And again, we found that there were basically no effects of rooting those cuttings under high pressure sodium lamps or under LEDs. We basically found that they all came into flower around the same time, and in terms of height, they were all about the same height. So if you'd like to read more about these studies, you can download the various articles that we've written on my website, and I'll give you that address at the end of the presentation here. So we're going to move on to plug production, and basically here, very similar to the previous study, this is some work that Wesley Randall conducted. And what Wes did is he basically had a high pressure sodium treatment, and again, 100% red LEDs or increasing amounts of blue light. We used a different type of LED in this study, and here we increased the light intensity to 100 micromoles for 16 hours a day. The temperature set point here was 70 degrees Fahrenheit, and the plugs were under the various supplemental lighting treatments for 28 days. So we looked at a wide variety of species. Unfortunately, due to our time constraints today, I'm only going to focus on one. But if you want to find out about each of the various species, again, we have an article that we published in Hort Science that talks about all of the various species. Here are the peak wavelengths for the various LED treatments and the high pressure sodium lamps. And here we have our Petunia Blush Blue. So again, under supplemental light, sunlight and HPS lamps, and then our increasing amounts of blue light. One of the things that obviously you can tell is that those links that were produced under LEDs are much more compact than the ones that were produced under high pressure sodium lamps. And we can definitely see that in this figure. You can see that statistically, those those plugs under LEDs were much more compact. And really, when we provided blue light, we found that they were compact. In terms of the stem caliper, we basically found that there were no statistical differences. When we looked at the root dry mass, which is obviously important because if you have more root dry mass, that means that plug is coolable, so it's ready to be transplanted. We basically found that there were very little differences in terms of root dry mass. We did have the highest root dry mass under our 85 red and 15% blue, and that slightly decreased when we increased the amount of blue light. And again, in terms of shoot dry mass, we had slightly higher under the high pressure sodium when we compared that to those seedlings under 70 red and 30% blue. We then, again, in this study, wanted to grow the plugs under a common environment, so they were transplanted into 4-inch containers, grown at 70 degrees constant temperatures, and we wanted to maintain a daily light integral of between 10 to 12 moles. So we used basically ambient solar light and high pressure sodium lamps. So this is 3 weeks after transplant, and you can see that there's little to no difference in terms of time to flower. So there were basically no statistical differences. And again, the height or stem elongation at flower, there were no differences. What can we conclude from this? Well, these are comparable to high pressure sodium lamps in terms of use for cutting propagation. There were no detrimental or beneficial effects of using LEDs, so they're quite comparable to the HPS. LEDs are suitable for providing supplemental light for plug production with some benefits compared to HPS, and some of those are that the plugs can be more compact. We also find that they tend to have more chlorophyll, so they're darker. Pigmentation tends to come out, and that the finished plant quality of most of the species was not significantly influenced by supplemental lighting from either high pressure sodium or LEDs. So again, we have some articles that you can download on our website, and we're going to move on to sole source LED lighting. So in other parts of the world, we're starting to see a large increase in sole source lighting, indoor production of either young plants, vegetable, primarily leafy greens, and this is just 2 examples of that. And so our next study, what we're going to do is we're basically going to compare plants produced under supplemental light in the greenhouse to plants produced indoors under sole source lighting under a very similar daily light integral. So we're looking again at plug production here. We wanted to really quantify those differences between seedlings that were grown under our ambient light levels. In Indiana, during peak production, we're about 6.5 moles of light. And of course, then we used our supplemental light as well as our sole source lighting. Our plugs were grown at 73 degrees Fahrenheit, and we maintained a daily light integral of about 10.5 moles. So that's again with our supplemental lighting. And our greenhouse supplemental lighting treatments consist of high pressure sodium, a plasma lamp, a LED module that provided 87% red and 13% blue light. From those previous studies that I talked about, we found that about 85-15 was a good combination in terms of red and blue light, so we chose this production module that had very similar. And then again, we had our ambient treatment, which had no supplemental light. In terms of those supplemental lighting treatments, lights were on for 16 hours, and they provided 70 micromoles of light. Now we're going to move to the indoor production, where obviously we had to increase the light intensity in order to achieve that 10.5 moles of light. So we had we provided 185 micromoles of light from 2 LED treatments, the 87 red, 13 blue, or 70% red and 30% blue. We've worked with 5 different species in this study. And here we have our plasma light treatment. So you can see that in terms of the light, it appears to be white. It's basically very similar to the spectrum of sunlight. And we have our high pressure sodium, as well as our LEDs. And then we have our 2 sole source LED treatments, which were in a growth chamber. So again, here we have our various spectral quality for our various supplemental lighting or sole source lighting treatments. And here we're looking at the midnight. So when we look at these figures, I just want to point out that all of these plants, the first 4, were produced in the greenhouse under either ambient or supplemental light, and the last 2 plants were produced indoors under sole source light. So we can obviously see that there are quite a few differences. In terms of height, that's probably one of the most pronounced. We see that when we used LEDs, those seedlings, Petunia seedlings, were much more compact, and when we produced them indoors. And so one thing that I failed to mention for those of you that are not familiar with young plant production is, in terms of a propagator, really we want nice compact seedlings that are, have a nice or large stem caliper and increased root dry mass. So we can definitely see that after 28 days, those seedlings that were under plasma lamps were quite elongated, as we can see there, and the most compact ones were under sole source LEDs. What about root dry mass? Well, as we would expect, when we provide supplemental light, we definitely have an increase in root dry mass, with the seedlings under sole source lighting having the highest root dry mass. In terms of shoot dry mass, we definitely saw that shoot dry mass increased under supplemental or sole source lighting, but there were really no differences between those various treatments. Then we transplanted plants and grew them under a common environment here, 68 day, 65 night, and tried to achieve a daily light integral of 10 to 12 moles with the use of supplemental light. And here we can see that, again, these plants were basically propagated as plugs under greenhouse conditions or supplemental light, and then the last 2 were propagated under sole source lighting. In terms of the flower, Petunia is a long day, and so we were obviously producing this plant under short days in the greenhouse, so there was a delay in time to flower. We can see that our high pressure sodium treatment, there was no statistical difference, but it was a few days earlier. With our plasma lamps, we could definitely see that time to flower was reduced, and we can attribute this to basically from the heat that was provided by the plasma lamp. As the air or as the plant temperature increases, those plants tend to develop faster. We also see that under sole source lighting, there was a delay in terms of those seedlings that were produced under the 87% red and 13% blue treatment. And in terms of height, there were some differences, but really in terms of commercial production, not much. The conclusions that we can take from this study are that the quality of all seedlings under supplemental light was similar or greater than those seedlings that were grown under ambient light. So obviously, providing supplemental light, you're going to have a higher quality plug. And we also found that seedling quality of species grown under sole source lighting was similar or greater than that of seedlings grown under supplemental light in the greenhouse. The flower was similar for species, that's the mistake there, under both supplemental and sole source treatments, except for impatiens and Petunia, where we did tend to find some delays. And so supplemental and sole source are both viable methods of producing annual bedding plant seedlings. This is a study that we're actually currently conducting, so I'm going to show you some very preliminary data. And here we really wanted to quantify the effects of sole source far red light on seedling quality and subsequent time to flower. So in terms of long day plants, let's say we produce them as young plants indoors under sole source lighting, we wanted to see will far red light help in terms of their subsequent flowering so that there's no delay. So we're working with 3 different species. Here we have Coreopsis, Pansy, and Petunia. They're all long day plants. In terms of their environment, we're producing them under a 60, actually they should say 70 degrees Fahrenheit, and a 16-hour photo period. We're looking at 3 LEDs, so we're looking at the 87 red, 13 blue, or a treatment that has 84% red, 7% far red, and 9% blue, or a LED that has 74% red, 18% blue, 18% green, and 8% blue. And then 3 different daily light integrals. So basically, we can increase that daily light integral by increasing the number of modules. These are our results. We're basically finding that increasing the daily light integral can obviously lead to decreased stem elongation. We had slightly smaller stem caliber under the lower daily light integrals, and then higher daily light integrals led to greater root and shoot dry mass as we would expect. Again, with this study, we transplanted those seedlings into the greenhouse under a common environment, and this is what we found so far is obviously by providing those seedlings with far red light during the young plant stage, they're going to flower faster when we transplant them into the greenhouse. So with those seedlings under the highest daily light integral, and if there was far red light under the sole source lighting, they flowered about 6 days earlier, and under the medium daily light integral, they flowered 8 days earlier. Our preliminary conclusions from this study that's still ongoing is that we have higher quality plugs under higher daily light integrals, as we would expect. Light quality does not appear to have a significant effect within daily light integral on plug quality, which is a good thing, because one of our hypotheses was that by using that far red light that we would potentially have some excessive stem elongation with those plugs, but we're not seeing that. Earlier flowering for some species does occur when you have that far red, or the addition of far red in addition to the red and blue under higher daily light integrals. Again, we have articles on our website that you can download to learn more about the other species. And now I'm going to move on to some work that we've been doing with microgreens. This is work by Josh Garavak, who's a former graduate student, and then Josh Graver, who's a current graduate student. And the objectives of this study were basically to quantify, again, the effects of sole source LEDs of different light qualities and intensities on the growth, morphology, and nutritional content of Brassica microgreens. We looked at 3 different Brassica microgreens: kohlrabi, mustard, and mizuna. And we basically grew those under a hydroponic system with these fiber pads. Our environment within the grow chamber was 70 degrees during the day and 63 during the night, and we had 500 parts per million CO2. Again, we had 3 light quality treatments. They're the same ones that I talked about earlier, and again, we had 3 different daily light integrals ranging from 6 to 18 moles of light. So this is what our various treatments look like. This is what our kohlrabi look like. So as we move from left to right, you can see we increase our daily light integral, and again here we have our 3 different light qualities. I know it's hard to see from this photograph, so let's move on to the next one. But one of the things that you notice is that with increased daily light integral, we tend to have a much more compact microgreen. Now, it's obviously you can ask different people, and they're going to tell you which is better. Some folks would want microgreens that are a little taller. Others may want them a little more compact. In terms of the hypocotyl length, we can definitely tell that under lower light, those hypocotyls are much longer. In terms of light quality, not a whole lot of difference there. In terms of leaf area, we have a larger leaf area under lower light compared to under higher light. Now, this probably would make a difference, obviously, if you're producing microgreens and you're selling them either in terms of the size or by the weight. This may have, obviously, an effect on that. And the dry weight, however, we can see that you're going to have much more dry weight, or excuse me, percent dry weight under higher daily light integrals compared to under lower daily light integrals. And we also found that the relative chlorophyll content increased when you increase that daily light integral. We found that anthocyanins tended to increase when you increase that daily light integral. And at the higher daily light integral, when you had that green light, we actually had a slight reduction in total anthocyanins. When it comes to total phenolics, we found that basically there were no differences between the various daily light integrals and the different light qualities, with the exception under low light, where there was a slight reduction in total phenolics between the microgreens under red, green, and blue compared to those red, far red, and blue. Now we're going to look at another species. This is mustard. This is a species that definitely has more of a red foliage, and you can see that when you increase that daily light integral, you definitely increase the anthocyanins, or the pigmentation within those microgreens. Again, look at the side view. You can definitely see that they're much more compact when you provide more light. So some of the conclusions that we can get from this study are that biomass production increased with higher light intensities, with increased DLI that led to an increase in anthocyanin content. Lower daily light integrals may lead to increased carotenoid content. I didn't talk about that. Again, we're still analyzing some of that data, but it's showing us that lower DLI may lead to that. And of course, there's electrical savings when you grow microgreens or bedding plants under those lower light intensities. We're going to move to a completely different topic. This is some work that Garrett Owen has been working on. He's basically looking at the end of production lighting with red and blue LEDs to increase pigmentation of lettuce as well as some ornamental crops. One thing that we have noticed under both supplemental and sole source light is that when we increase the amount of blue light, we tend to have more pigmentation, so anthocyanin production. And so one of the things that we often notice in the greenhouse is that under low light conditions, purple fountain grass or Penicillium rubrum, and also red leaf lettuce, is that often that foliage color is light green, pale, not very aesthetically pleasing. So here we have a greenhouse that is producing purple fountain grass, and when I look at that grass, it's pretty green. And one of the reasons is that obviously we're producing this in the spring, but often growers have a lot of hanging baskets or excessive shade, and really the appeal of this grass is that it has purple foliage, and that's why consumers want it. So here's a close-up of what we often find in greenhouses where that purple fountain grass really is green fountain grass. Really, color is a key component in terms of consumer appeal. It influences and registers with their initial perceptions of product quality, whether it's a lettuce that they're going to consume or an ornamental that they're going to place in their garden. So color of plants is mainly attributed to pigments, those chlorophylls, carotenoids, flavonoids, and of course those anthocyanins. So research has shown that the addition of blue light significantly increased the concentration of anthocyanins within leaf tissue. So we came up with the idea of end of production supplemental lighting, where basically a grower could grow their crops, whether it was lettuce, purple fountain grass, or other foliage ornamentals, under the same low light conditions, and then right before that plant is either going to get harvested or shipped out, they would light the crop under the treatments that I'm going to talk about to increase the overall quality or aesthetic appeal for the consumer. This is the first study that I'm going to talk about. We basically received purple fountain grass, and you can see that it was very light green in color. We grew it out for 15 days prior to the production lighting. We wanted to simulate the daily light integral that we would find in a commercial greenhouse because of those hanging baskets. And what we found is typically it's about 6 moles, which is really low when you think about the light levels that we actually have in March. We then basically finished the crop under our end of production lighting. We had a 16-hour photo period and a daily light integral of about 9 moles of light. And here's the various treatments that we had. So we had basically our control, which was basically no end of production lighting. We just wanted to maintain a 16-hour photo period because all of the other treatments had 16-hour photo period. And again, our LED treatments were with red on the left and blue on the right. So we had either a 100% red treatment at 100 micromoles or a combination of red and blue at 100 micromoles, 87% red or 13% blue, or increasing intensities of blue light from 25 micromoles all the way up to 100. For our various LED, so this you can see is our lettuce on the top and our purple fountain grass on the bottom. And again, what these our treatments look like at night. With our purple fountain grass, this is basically what the plant material looked like. So it's getting close to being what we would consider marketable. We then moved them into our various end of production supplemental light treatments, and this is what we found 7 days after the initiation of those. So our control, which is basically just receiving 16 hours of photoperiodic lighting, you can see there's very little purple pigmentation, even with our high pressure sodium. So we chose 70 micromoles because that's about the industry standard in terms of supplemental lighting with HPS. And again, you don't see very much color. With 100% red light at 100 micromoles, you can start to see some pigmentation. Now, when we increase, or when we have 50% red, 50% blue at 100 micromoles, you definitely can see an increase in pigmentation. 8713, not so much. And again, as we increase the amount of blue light all the way up to 100 micromoles of blue light, we again see that nice dark purple foliage. 14 days later, you can definitely see that 50-50 and 100% blue at a high intensity really got us to have that nice purple foliage. When we take a closer look, you can definitely see this. This is after 21 days. So look at the foliage under 100% blue light at 100 micromoles, how nice and dark it is, and even at 50-50 and at 100% red. What about the flowers? So again, with Petunia Rubrum, when we provided a high light intensity of blue light, look at how the flowers were much more purple in terms of the color. Excuse me. Here's another side view. You can definitely tell that 100 micromoles of blue light, we have a actual purple fountain grass, and even when we have the combination of red and blue or red light alone at a high intensity. We also looked at other ornamental species. So this is a geranium. It's called black velvet, primarily grown for its foliage color, but you can definitely tell that we can get that really nice color that we would find outdoors, but under our greenhouse conditions when we had or when we grew these crops under end of production lighting, either providing 100% red light, 100% blue light, or a combination of the two at high intensities. Let's look at red leaf lettuce. So here what we did is we selected 4 red leaf lettuce cultivars. These are all obviously red, and this is basically the size they were and their color right before we placed them into the various LED treatments. So here we have lettuce Cherokee, and with lettuce we can see that the effects are much more rapid than they are with the ornamentals. So after 5 days, we can definitely see a nice color with our red and blue treatments at 100 micromoles, and especially right here with these 2 treatments, or at the 100 micromoles of blue light, we can definitely see again as the number of days under the end of production lighting increases, we really start to see a nice increase in the overall color of Cherokee. let's move on to Vulcan. Again, very similar results for that high intensity, but especially when we have that blue light, we have an increase in anthocyanins and, of course, that nice pigmentation. Here we have Ruby Sky. Look at how nice this plant looks after 14 days, but really the crop could be marketed after 5 to 7 days. And here we have Magenta. This is probably the least responsive of the cultivars that we worked with, but again, after 14 days, nice color under a combination of red and blue light, and even the 100% blue light at 100 micromoles. What are the conclusions that we can take from this study? Well, in terms of the purple fountain grass, 7 to 14 days of end of production supplemental light, providing 100 micromoles of either red or blue light, 50-50. Red or blue light alone was effective as it promoted purple pigmentation of the purple fountain grass as well as the black velvet geranium when those plants were grown under relatively low greenhouse DLI. With the lettuce, 5 to 7 days of the same treatments were effective as they promoted red pigmentation of the 4 cultivars that we looked at. So we're going to move on now to photoperiodic lighting. We've looked at both photosynthetic and photomorphogenic lighting, and now we're going to move on to photoperiodic lighting. And this is some work that Garrett Owen and Christine Adamek worked on. And in terms of horticultural crops, they can be classified as either day neutral, short day, or long day. And typically, most growers will provide photoperiodic lighting strategies for long day plants. That's either night interruption, where basically the night is interrupted between typically between 10 p.m. and 2 a.m., or day extension lighting, where basically right before the sun goes down, these photoperiodic lights are turned on to a certain, let's say maybe 16 hours or 14 hours, depending on what the grower is trying to achieve. Typically, growers would use incandescent lamps, which emit low red light and a high amount of far red light. One of the problems, however, with incandescent bulbs is they're very electrical inefficient, only about 6% effective at producing light. So that's why incandescent bulbs are so warm, is that they're producing a lot of heat. They have a relatively short lifespan, and of course they've been phased out of production, so it's going to be much harder for a grower to find an incandescent bulb. Compact fluorescent lamps emit a high amount of red light and a low amount of far red light. So alone, it doesn't provide an effective flowering response because again, far red light is required in terms of flowering. One of the drawbacks with CFLs is obviously their life is influenced by the number of on and off cycles, so that's going to reduce its life. And of course, they pose disposal issues because they contain mercury. Then some growers will use high pressure sodium lamps for cyclical lighting, so we can see in these two photographs whether that high pressure sodium lamp oscillates back and forth, or it can be attached to a boom, which then moves across the lamp. Obviously, this is energy intensive because high pressure sodium lamps require quite a bit of energy. They also emit radiant heat, and in terms of the response, those plants directly under the high pressure sodium lamp are, in some cases, might flower faster than those that are at the farther end. So now we have low intensity LEDs that have been designed to replace incandescent bulbs, so that basically they can screw into the fixture. They have a much longer operating life, and the spectra can be tailored to elicit desired plant responses such as flowering, or in some instances, stem elongation for, let's say, a cut flower. We looked at a wide variety of species, both long day and short day plants. You're probably thinking, well, why would you want to use photoperiodic lighting on a short day plant? Well, let's say we want to keep it vegetative in terms of keeping it vegetative before we want to take it to market. So these are the different species that we looked at. We had an automated black cloth system. the curtains were retracted to create a 9-hour photo period. So basically from 8 to 5, these plants receive natural sunlight. We also used high pressure sodium lamps to really increase our daily light integral because we want to obviously produce a high quality crop. We had a greenhouse air temperature set point of 68 during the day and 65 at night. And we looked at 2 different daily light integrals. So one of 5 to 8 moles, so this is basically spring production, and another of 13 to 15 moles, which we would be more of a summer production. And one of the reasons that we chose to look at 2 different daily light integrals is that the LEDs that I'm going to show you, some contain just red and white light, while others contain red and far red light. And so we were hearing that growers in, let's say, the Pacific Northwest or in the South could use only the red and white, and they would get a flowering response, where growers in the Midwest or northern parts of the U.S. would have a delayed response. Again, we used day extension for our experiments. We provided about 4 micromoles of light from these photoperiodic lights. So one of the things that's a good take-home message is, with photoperiodic lighting, requires relatively low light intensities compared to the photosynthetic and photomorphogenic lighting that I talked about earlier, where we were looking at anywhere between 70 to 100 micromoles. So the 2 LEDs that we looked at were the red, white, far red, which were on for either 1, 4, or 7 hours, or an LED that just contained red and white light, again for 1, 4, or 7 hours. So here's our treatment. You can see the 2 bulbs. Because we're using these small chambers, you can see that we had to use a screen in order to get the lower light intensity that a grower would achieve with their photoperiodic lighting. Here's our results. So here we have pansy, long-day plant, relatively low daily light integral of 6 moles of light. And here we have our plants under the red, white, and far red LEDs. And here we have our red, white LEDs. And what's the thing that we notice? Well, under a 13 and 16-hour photo period, we have flowering when we have that far red light under a low daily light integral. When we look at those under red and white light, the time to flower obviously increased. What is this? About a 16 days, so 2 weeks here and 17 days here as well. So it's quite a big difference in terms of flowering. So what's something else that we found? Well, obviously one of the drawbacks of far red light is it induces stem elongation. So when we look at these plants compared to these plants, obviously when we look at the data, there was a difference in terms of their height at flower. Here we have Petunia, another long day plant. We can definitely see that we had a relatively low daily light integral of about 6 moles of light. And in terms of flower, look at those plants receiving that far red light. They flowered nearly, what is this, almost 3 weeks faster when we had that far red light than when we had the red and white light alone. However, when we look at the height, look at how much taller these are. Obviously a significant difference in terms of the overall height. And here we are looking at Snapdragon, again, another long plant. When we produced grew our Snapdragon under those red, white, far red LEDs, they flowered much faster than when they were under that red and white light. Again, elongation, obviously, or that height at flower, they're much taller under far red LEDs, but they flower much faster. So it's all dependent on obviously what you're looking for. This coming summer, we're going to be looking at the highlight. So basically looking at higher higher daily light integrals. And we've already done some preliminary studies and have found that under higher daily light integrals, you can actually eliminate the far red light. And so some of our conclusions are that under low DLI, low intensity LEDs with far red light obviously increase stem elongation, but they hasten time to flower of those long day plants. Under lower daily light integrals, under low DLI, low intensity LEDs without that far red light, had reduced stem elongation, but there was obviously a delay in terms of time to flower, and it varied with the various species. Under low daylight, day extension with white far red photoperiodic lighting hastened time to flower, obviously producing a market-ready plant. Obviously, it was also very species dependent, but it promoted stem elongation. I wanted to let everyone know about our LED website. So we, along with several other universities, received a grant from the USDA SCRI, and some folks are working on vegetables, others on ornamentals. And so this is the website where you can find all of the information of the various studies, the papers that have been published. We're also going to have an LED symposium next week in Tucson, Arizona. And there is still some space available online. I know I'm looking forward to heading to the Southwest and get away from the Midwest cold. So this day-long symposium, we're basically going to talk about all the LED research that has been done at the 4 different universities. And with that, I would like to thank the USDA, private horticulture and LED companies that have supported a lot of the research that I've talked about today. Obviously, today I did not have time to talk about everything that we've done, but I just wanted to give you a snapshot of the various photosynthetic, photomorphogenic, and photoperiodic lighting treatments that we've done over the past 4 or 5 years. And with that, I would like to thank Phillips once again for sponsoring this webinar, and we have plenty of time for questions.
Roberto, can you hear me now? Roberto.
Yes, I can hear you.
Okay, thank you. So everyone, there is plenty of time for questions. So if you have a question, type it down in the chat area of the control panel, and then we'll add those to the queue, and then we will, I'll start reading them off. So let's see. I'll go to the top. Man, there's more questions here than I saw. Okay, the first question, Roberto, is from Paul, and he says, for seed germination under SSL, LED lighting, and using LED lighting, I believe, what red to blue mix is best or optimal?
Okay. So one thing, again, because I was trying to include everything that we've done, I didn't talk about the fact that the seedlings were actually germinated in the greenhouse, and once they had started to emerge, then we transferred them into the sole source lighting. So they weren't necessarily germinated under the LEDs, but they had started to emerge from the substrate. In terms of the young plants, you know, it's all very dependent on what you're looking for. Obviously, I said most propagators want a nice compact seedling that has a nice large stem caliper, root dry mass. From the studies that we have done, I would say that 85% red, 15% blue is a nice combination. As I said, we're doing some ongoing work now with including either green light or far red light, and if you're producing long day plants, those may be good treatments. Right now, I would say that we need to wait a little bit longer to see if you know, there's any negative effects on certain species in terms of that stem elongation.
So then so you're finished with that question?
Yes.
Okay. So then the next question comes from Ron. Would the different spectrum of red and blue LED between the research and production module have an impact? So when I read that, I'm not quite clear exactly what's being asked.
So can you repeat that question again?
So the question from Ron is, would the different spectrum of red and blue LED between the research and production module have an impact?
If there were an impact, I would expect it to be relatively low. You are correct. Between the research and the production modules, there is a slight difference in terms of the peak wavelengths within the red and the blue, but I would expect it to be relatively low, if any.
Okay. There was a question from Ziu, I think, if I'm pronouncing it right. Is it possible to get a copy of your presentation with the bar charts included? There's a PDF that you can obtain right now under the materials area. You can download it, and through Monday on the e-GRO website at e-gro.org, under the webinar tab at the very top, there is you can obtain a copy of the PDF there. When we repost the webinar, the PDF will also be there, but that's when we'll post the webinar recording in about 10 to 14 days. So, okay, then I've read that one. Paul had a second question. What SI LED would be best to maximize foliage on rosemary 4-inch in production?
So which supplemental LED for 4-inch geranium, you said?
To maximize foliage on rosemary in 4-inch production.
Well, it's obviously what is it that you're looking for? Are you looking for the increased growth? Are you looking for certain parameters? It's kind of a question that probably, if you want to contact me, it would be easier to go through the various scenarios of what we have found with the various LEDs. So, yeah, I would say that it's probably best just to contact me via email, and then we can go back and forth and see what it is that you're ultimately looking for.
Yeah, I think the first slide of the presentation had Roberto's email address on there, so you can use that to give him a question. So the next question. Go ahead.
I can post it. I can put it in the comment section here. I can put in my my email address.
Yeah, that'd be great too. Okay, sorry, someone else asked. Questions move around on me. So Blas, I think I'm saying it right. Do you also have some research with laser lights?
We do not have, we have not conducted any research with laser lights.
Okay. So Joe Erica asked, is there a standard or optimal height between LED lights in the plant?
With about high pressure sodium lamps, we often, we have to place those lamps quite a nice distance away from the plant material because of the heat. Now with LEDs, we can place those plants relatively close to the plants because they produce relatively little heat. And in terms of that that's the great benefit about multi-layer production, is you can really have the LEDs close to the plants and with no detrimental effects. Obviously, you can't get too close because you want to have nice uniformity in terms of the light intensity between all of the plant material. So it's dependent on the output of the LEDs, the intensity, obviously the intensity that you're trying to achieve.
Okay. So there's a question here from Steve. It's two parts, and I'll answer the second part. Is this recording going to be available for download and or viewing? The answer is yes. We'll have it at the e-gro.org website under the webinar tab, and we will post that within 10 to 14 days. The other question, Roberto, that Steve asked was that for the microgreen trial, were there nutrient supplements added?
Yes, there were nutrients added at every irrigation.
Okay. You remember what they were, or standard?
Off the top of my head, I know Josh Garavak is on the webinar. If he did the study, he probably could tell us the exact amount. We use a Hoagland solution, but I cannot remember the concentration off the top of my head.
So if Josh wants to give that answer in the chat area, that would then be provided. That's an easy way to be done if you remember it off the top of your head. So sorry to put you on the spot there, Josh, but you're the expert. So the next question comes from James. How can I measure PAR accurately under IED lighting to integrate with my control system?
Okay, this is a great question, and this is a question that I often get from growers. With LEDs, you cannot use a foot candle meter. You're going to have to use a quantum meter or a spectroradiometer. Obviously, a spectroradiometer is they're quite expensive, but if you want, with the spectroradiometer, you can determine obviously the light intensity of the LEDs as well as the wavelengths. So, you know, it's a big investment, but a quantum sensor such as a Light-core, those have been calibrated for LEDs, and so that's what I would recommend.
Okay, so Ron has another question. Is it still right using PAR, in parentheses PDFD, while measuring LED intensity, or should I be sorry, just moved. Sorry. Every time a new question comes, the questions move on me, so I have to move back up to find it.
I believe I know what the question is asking. So if you are using LEDs that are within PAR, so 400 to 700 nanometers, you're great. But if you have LEDs that have far red light, then you obviously are going to have to measure within the far red region. So knowing that your sensor is calibrated or has the capability of measuring, let's say, between 400 and 800 nanometers is important.
Roberto?
Yes.
Okay, so, okay, sorry, I thought we lost you for there. Okay, I'll ask that one. There's another one we'll go over later. From Dell, have you done any studies in terms of shorter term exposure to far red lighting? Example, only at the end of the day rather than all day as a solution to not having stem elongation but retaining flowering benefits.
Yes, we have done, with plugs, we have done end of day lighting. So we've looked at red light, blue light, and far red light. And one of the things that obviously we know is that at the end of the day is when there's quite a bit of far red light as the sun is going down. So in terms of stem elongation, that's, you know, can obviously trigger stem elongation. So our thought was, what if we end the day with red light, or especially blue light, which is known to produce compact plants? Unfortunately, with the plugs, we did not find, you know, they were compact for the first week or two. However, when you think of producing plugs, they're very close together in a very tight competitive environment. And so after those 2 weeks, basically, they were all the same height, whether they were under far red, or blue light. So we may pursue this in the future. Again, in terms of flowering, that's a great question. If you could only, let's say, provided far red light at the end, would we still receive or find a flowering response? We don't know. So that's something we may do in the future. As you can see from today's webinar, there are so many possibilities of things to do with LEDs. We just, you know, it's going to take us a long time to conduct these various studies.
All righty. A question from William. How would you compare LEDs tested with white fluorescent lamps for the sole source lighting?
How would we compare them?
How would you compare LEDs tested with white fluorescent lamps for sole source lighting?
Okay. At Michigan State University, they have conducted studies looking at a wide range of colors with LEDs and then compared those to fluorescent lamps. So the research has been done. If you want to find out the results, you can go to the MSU Floriculture website under the research tab, and they publish some articles, I know, in Greenhouse Grower magazine.
All righty. As I'm working down the questions, I do see that Roberto did post his email address there, so that is there, and he also provided a link to their website at the Purdue server, so if you want to look at that. So we're down to a question from Carlos, and I know it is up at 2 o'clock, so for those of you who want to go ahead and get off, you're more than welcome, but I think we can spend the next, I think there's only 2 or 3 more questions. We're going to go ahead and get those answered while people are still online. So the question from Carlos to you, Roberto, is what do you expect by adding green light to your later experiments? And why did the blue light promote more compact plants?
So one of the things I failed to mention is that with that treatment, it basically contained red and blue light. Now, when we look at the light, it actually is white. And the reason that it's white is that they put a phosphor coating over the blue LED. When we use that spectroradiometer, we actually see a small peak in the green region as well as in the blue. So we didn't necessarily add green light, it's just that's what the plant and obviously the spectroradiometer perceives. With blue light, that's one of the nice things with blue light is that we do find that plants are more compact under blue light. I hope that has answered your question.
Okie doke. And did you, it's okay, he had both parts, right? And why did blue light promote more compact plants?
I mean, that's just a fact in terms of blue light.
Okay.
Is that typically plants are more compact.
Okay. And there's a question from David. Are you still planning to do studies on whether LEDs can have an impact on the nutrient vitamin content of microgreens?
Yes, so we are currently, we've done those studies and we're currently analyzing the data where we've worked with a collaborator at the University of Tennessee, and we're looking at chlorophylls, carotenoids, and luteins, and basically, yes, trying to determine if the various light qualities or light intensities and how they may affect compounds that are beneficial to human health, as well as look at if LEDs affect the micro and macronutrient status of crops.
Okay, so right now it's the last question. So if anyone else has, well, I also want to put one, there's 2 more left. James asks, where can I find those calibrations?
Oh, in terms of the, I believe he's talking about the light meters. So what I would suggest you do is if you have a light meter, I would contact the manufacturer and ask them, has the meter been calibrated or is it calibrated for LEDs? Some meters have what says electric lamp, sunlight, but it may not be calibrated for LEDs. So I would say definitely contact the manufacturer. They can tell you. They may also tell you that the meter may overestimate or underestimate a certain light quality, and so then you may have to account for that. But it's I would say the manufacturers can tell you that.
Okay, so right now it looks like the last question, unless another one comes in. Derek asks, is there any benefits to having green LEDs added to your lighting system?
So again, Michigan State has done research using orange light, green light for sole source LEDs, and I mean there's no benefit and there's no detriment. So you know, it's why have we focused, or why, I shouldn't say we, but why have the LED manufacturers focused on red and blue light? Well, in terms of photosynthesis, those are the two wavelengths that you tend to have peaks in, so that's why they focused on those LEDs. But no, there's no benefit or detriment.
Okie doke. All righty. Well, that looks like it's the last question that was in there. Roberto, I want to thank you for an excellent presentation and sharing your thoughts on LEDs. We'd also like to thank again Philips for sponsoring this webinar. So the webinar will be recorded. It will be posted on the e-GRO.org website under the webinar tab, along with a PDF of the slides, so you can look at that information later or share it with other people that might want to watch it. So we thank you for attending the webinar, and hopefully you might find some of the other webinars coming up also to be beneficial to you. So thank you very much for attending.
Thank you, Brian.